Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosland, online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Dr. Lauren Sierkstra from Thermo Fisher Scientific.
Hello, good morning everyone for listening to this webinar today. Today we'll be discussing, let's say, the solutions Thermo Fisher Scientific has for developing specific custom chromatography resins. And just as a reminder for you as an audience, right, that Thermo Fisher Scientific actually has got leading capabilities for every step in the bioprocessing workflow. So from cell culture media and development up to scale-up and production, harvest and collection, as well as pharma analytics to actually measure, let's say, contaminants in your process. We as well have a purification resins, which actually help you in purify your final product. And with that, we've got a couple of lines of products. First is our Capture Select resins, which is currently the largest portfolio of antibody resins for the purification of virtually any antibody format, but as well for biosimilars, biobetters, and viral vectors. Our porous resins, which is a whole range of ion exchange resins, as well as HIC resins, but as well with, let's say, added affinity resins, for example, for messenger RNA. And these products are actually all used in many commercial and late-stage processes, being produced in state-of-the-art manufacturing facilities, whether it is the affinity ligand production, which we have in the Netherlands and Lithuania, as well as, let's say, our large-scale resin manufacturing capabilities within the Bedford site in the US. Besides our portfolio, which we have, let's say, for types of and classes of molecules, we as well offer custom resin development programs, which are tailored towards specific customer molecules. And the reasons why we've actually invested in the technologies behind this is that if you look, let's say, 10, 15 years back, the predominant molecule class which were basically in clinical development were antibodies and as well simple, straightforward monoclonals. What we've been seeing over the last 5 to 10 years is a shift in the pipeline of these global biologics entering the clinic. So although antibodies is still a predominant part, like say 31%, we as well see a lot more recombinant proteins, cell therapies, gene therapies, and of course nucleic acids like messenger RNA, let's say, hitting either the clinical or the commercial stage. All of these new modalities have their specific purification challenges, and actually that drives a need for additional tools. So that's why we've invested in specific products targeting classes of molecules. So for example, we've got the leading product for adeno-associated virus purification. We've got the leading product for messenger RNA purification through our oligo-DT resin. A whole variety of antibody resins to purify novel antibody formats. And again, that is then talking only about specific classes, right? Why do you want to, and why would you want to pursue a custom resin? One of the reasons could be, right, that although, of course, we've got a big portfolio of affinity and polish and ion exchange resins, there might be no suitable chromatography resin which actually exists, which can purify your product. It might be that the current resin, which is available on the market, actually lacks some specificity. So it might actually provide some additional resolution requirements which current products do not offer. Could be that the current process is actually not scalable. So we work with customers which, although they have a process, in the end cannot hit, for example, a host cell protein removal, which is needed to make it into the next clinical phase. Of course, there are specific molecular requirements. So it might be that available chemistries restrict the design space of your process, or limit certain operation conditions. And finally, if you have a, let's say, non-platform process, that usually is composed of multiple steps to purify your product. And actually, by employing a custom resin, that might lead to a simpler process with higher yield, reducing in a cost of goods for your process. So although we've got a whole range of products which suit specific classes, there might be specific cases where a custom resin is really worthwhile to implement in your process. So for one of the areas we have developed over the last 10, 15 years is a capture select custom ligand and resin development program. This specific, let's say, product and technology we've actually, let's say, set up in order to simplify biomolecule purification. So if you look to this graph and you take a look at the left-hand side with a typical, let's say, process flow, you can see that when you have non-affinity capture, you usually need multiple processing steps to end up with a pure product, right, which can actually be used in clinical trials. And with every step, and if you then take a look at the middle graph, with every step, of course, in a process, you'll have a yield loss, right? And even if you have a pretty good yield, step yield of any of these steps, let's say 85%, if you have 4 steps in a process, which is not atypical for a complicated molecule, you'll probably throw away half of your product during downstream processing. So all of this value, which is actually produced in upstream, half of that value is basically lost by the amount of process steps you're having. So the technology we've developed actually can shift that. So it can shift, let's say, processing from the left in this graph to the right in this graph by having simple affinity capture, usually followed by 1 or 2 polish steps, which will provide you with a high yield and a reduction in the number of purification steps. It will as well, of course, develop your and reduce the bioprocess development time, because the more steps you need to implement in your process, the more time it takes to develop, the more time it takes to validate as well. So in the end, incorporating an affinity capture step in your process, will reduce the time to market and your overall cost of goods. And this is specifically why we've developed the capture select affinity technology. So the affinity technology we have developed is based on antibody selectivity. So what we're using are called, let's say, VHHs, which are small antibody fragments composed of, let's say, 3 CDRs and 4 framework regions, only 15, so 15 kilodalton in size. And through our unique screening technology, we can include target specificity, mild dilution, as well as stability of the affinity resin. Key here in a lot of these type of, let's say, applications is, of course, target specificity, but as well mild dilution might be something which is really needed for, let's say, specific targets. All of our VHHs, which we manufacture, are based on our platform technology production, which is in yeast, so baker's yeast, Saccharomyces cerevisiae, which results in a completely scalable and animal origin-free production process. We've taken more than 40 of these products through, let's say, upscaling, and our products are currently in commercial purification processes. So, for example, for adeno-associated virus, antibody fragments, recombinant proteins, a lot of our products are in commercial purification processes. And since the last 15 years, we've got a proven, robust custom ligand development program to actually enable our customers to, in the end, have a scalable affinity resin solution. So this is the basic principle of affinity capture, which you probably all know. On the left-hand side, you can see the bind, wash, and elute. So basically, we design our ligands to be highly specific to the molecule of interest, which means that once immobilized on a appropriate resin, so an appropriate solid support, we can capture the target of interest and not the impurities which are in your process. by a simple washing step, you'll actually improve the purity because you're going to wash away the impurities. And then finally, you will elute the product from the affinity resin. Again, very straightforward process. And if you take a look at the gels, which are more or less in the middle, you can see 3 examples of 3 pretty dirty feed streams at the top, one for factor 8, one for antibody fragments, and one for AAV. And you can see that from the pretty dirty feedstock, with a simple wash and elute, you can get your highly pure product, actually in this case in one single step. Of course, you will need polishing additionally as well to reduce the final loads of some contaminants. But in principle, you will get, just like you're used to with, for example, protein A for a monoclonal, you'll get high purity product out of this. And it is really independent of the molecule you're making. So over the years, we've been introducing affinity capture for mainly all biologics classes. So we have a full range of products for antibody-derived therapeutics, whether you are, let's say, using antibody fragments or bispecifics, or the protein A site has been engineered out of your monoclonal. We have got a full portfolio of products to actually help you in getting a true platform process for your anti-derived therapeutics. We've got a full range for products for biosimilars, mainly actually the hormone family, because they are notoriously difficult to purify. Viral vectors, we've got the leading product for AAV, and vaccines and recombinant proteins, for which we've introduced the CTAG as a platform. And again, as said, all of our products are currently in commercial purification processes. So the development path we have to actually start from scratch, and this is really starting from scratch, from target to final resin, which is suitable for GMP processes, is a platform 5-step process. And that 5-step platform process will, in the end, result in a resin which is scalable and suitable for GMP processing. So if you take a look to the left-hand side, is what we call work package 1, which starts off with library construction. So we would take a target and would actually make a library through immunization. That library construction, because we have been working with the technology for around 20 years, we do have a lot of pre-existing libraries. So it might be that the target of interest is already in one of our existing libraries. If not, we would basically start with the immunization process, and after, let's say, the immune response would make a monoclonal library of all available binders within the immune system. The nice part is that we can as well work with more multi-target libraries. So, for example, we work with customers, and we are working with customers who basically have provided us with their pipeline. We immunize with their pipeline, and we can generate then one library which is composed of, let's say, monoclonals which are derived against any of the targets which is let's say, in there. So that is step 1, and we'll test, of course, the library construction, whether we are seeing a good response, a polyclonal response, and if that is okay, we're going to progress to the next work package, which is called work package 2. Work package 2 is really critical and key, and this is as well where we've done, let's say, most of and the majority of our improvements over the last years, right? So in work package 2, we're going to do the lead screening. So we're going to take the monoclonal library from work package 1, which is around 10 to the 9th different, let's say, monoclonals. And after a couple of rounds of, let's say, phage display, we're going to work in a high throughput mode in a label-free system. And we're going to analyze 200 different clones on, let's say, ligand kinetics and specificity. As said, that is in a high throughput fashion. So we're going to take the 200 clones and analyze them. Does the ligand actually bind to the target? Is there, let's say, enough specificity in the ligand which is going to be identified? What are the binding kinetics? So what is basically the on and the off rate? We can check, let's say, up to 20 different elution conditions in the system like this. So basically really making, let's say, uh, the resin in the end suitable for the product you need to purify. And we can check for ligand stability, which is of course finally as well needed to make a robust resin. So this step is really key to basically, let's say, end up with a good resin. Now, after work package 2, we're going to take 6 monoclonals, so monoclonal antibody fragments, so the VHHs, the 15 kilodalton proteins, further into what we call work package 3. And work package 3 is where we'll test the prototype ligand on a resin. So we're going to immobilize the ligand onto a resin, and we're going to build the prototypes and do the actual screening of those prototypes. In this step as well, we'll take into account ligand scalability to ensure that we can finally seamlessly upscale the product. We're going to check whether it indeed fulfills the criteria of the one-step purification, and we're going to check the elution profiles as well as ligand stability. So this is usually done in work package 3, and I said that will basically determine the final 2 ligand candidates, which are then taken over to work package 4, which is the final lead selection. In work package 4, we're going to upscale the ligand and actually test the lead prototypes as well with feedstock, and we'll evaluate it under process conditions. Finally, if work package 4 is successful and we will end up with 1 lead candidate, we can then seamlessly move into work package 5, which is to make that final resin suitable for GMP processes. That final resin is then made with 3 by 5 liter validation batches. Of course, it can be higher as well, but the minimum is 3 by 5. We'll develop a leakage ELISA for the ligand, of course, something which is needed for a regulatory submission. And that is actually the final close-off of the program, develop a regulatory support file. Again, this is a unique stage-gate service to develop tailored affinity solutions. And to be clear as well, right, the whole program is usually de-risked after work package 2, right? So if we see a good immune response in work package 1, we take it further into work package 2. And after work package 2, the program is fully de-risked. So we at that point know that we can deliver that affinity resin at the suitable scale in the time frame needed to go into a GMP manufacturing campaign. How we specifically work with customers in this case is there is right from the start a nice input file by the customer, which will be turned into an output file and a project proposal. It's nicely stage gates between the work packages, right? So we've got nice milestones determined to progress yes or no. And there is, of course, a continuous interaction with the customer at that point to ensure, right, that let's say every step is nicely monitored and discussed. Within work package 3, people can already have access to these prototypes, right? So we'll test them ourselves, but we'll as well have collaborators and customers test them. As well as in work package 4, there is already research use resin available for customers to do, for example, some process development. So it's not needed to, of course, wait until the full GMP validation batches are ready. So this is our stage gate process, and actually every product we've made, like say all of the 40 plus, have all gone through this stage gate processes. So we know exactly the timing and know exactly, when we can deliver the products for its further use. So, of course, really important is to, because it is all great that you can make a prototype, but it's of course important that we can as well make the final product at scale. So for that, we've got a certified manufacturing facility in Naarden in the Netherlands. It is ISO 9001, and the capacity of this facility is quite big, right? So we've got 215 cubic meter fermenters, which actually are operating together with the associated downstream processing. With respect to assurance of supply, of course, we have our cell banks stored at 2 different locations. We actually have manufacturing redundancy in Naarden, Netherlands, but as well in Vilnius, Lithuania, which is as well a Thermo Fisher Scientific-owned facility to actually make these type of products. And we've got 3 manufacturing facilities for chromatography resins. Finally, of course, we adhere to let's say, all quality and regulatory requirements to ensure that these products, of course, can be used in a GMP process. And for that, as said, we make available our regulatory support documentation through an online tool, which can be used for EMEA and FDA filing. So, as said, on the left-hand side, that's a picture of our Naarden, Netherlands facility, operating since 1997. Extensive know-how on manufacturing processes, specifically in Saccharomyces cerevisiae, completely animal origin free and fully, let's say, dedicated to llama single domains. It's ISO 9001, regular customer audits, of course, and as I've said, we have got additional processing, which we have as redundancy as well as main production in Vilnius, Lithuania. Then there are two resin sites because once we make a custom resin or a final product, we have a choice of two backbones, so two resins as a platform. We've got the agarose platform, which is through our partner on the Isle of Man, which is called Astrea, formerly Prometic in the UK. We have an OEM partnership with them since 2013. Manufacturing capabilities is scalable up to 300 liters for these affinity resins. And again, as well, ISO 9001. On the right-hand side, a picture of our Bedford, Massachusetts, USA location, where we make the porous resins and as well will do the immobilization of the ligand on the porous resin. Manufacturing capabilities, 250 liters, as well a highly, let's say, skilled R&D center. This facility is actually ISO 13485. And again, they mainly make, let's say, the porous chromatography resins there, which are fully scalable as well as, let's say, in numerous commercial processes. So, as said, we can, develop the products from scratch, but the guarantee is as well there that once the product has been developed and made, sourcing of these type of products is not an issue. So the track record we have in these type of products, and on this slide you can see on the left-hand side the different target classes we've worked on. So we've worked on proteins predominantly in customs, so about 75% has been recombinant proteins, antibodies 15%, viruses, gene therapy about 5%, and viruses, vaccines of around 5% as well. So really a full range of different target classes. If you then break the proteins up and move to the right-hand side of this slide, you can see the different protein subclasses we've worked on and where we've been successful. So a quarter of it have been recombinant enzymes, worked on blood factors, factor 8, factor 7, those type of product. Fusion proteins, so for example FC or interleukin fusion proteins, hormones, plasma proteins, and recently as well, and that is something we started probably about 3 years ago, cell surface markers, so CD for cell separation, as well as a small fragment there in toxins and others. Again, we've run more than 60 of these programs since 2003, and around let's say 40 of these products are now being used in clinical or commercial products. We have a very high success rate, over 90% in identifying lead candidates, meeting the predefined requirements which had been laid down when we started off in work package 1. Again, technology highly versatile, highly successful in actually generating these type of products. So besides, of course, our custom program, which we do, we continuously keep on innovating, of course, to keep track of new developments in the biopharmaceutical manufacturing space. So this is a snapshot of our pipeline, which are actually the products we either have in development or are close to being, let's say, launched as a GMP product. From top to bottom is basically as well our work packages, which I've discussed earlier. So if you go to the lower hand side, on the lower slide of this slide, you can see stage 1, which is basically work package 1, where we have got libraries. So for example, we've got a library against host cell proteins. And on the right-hand side, you can see a non-immune library, which we employ very often for viruses and vaccines. And if you then move up to the top, you can finally see our commercial processes. So on the left-hand side, therapeutic proteins, in the middle, the antibody types, and on the right-hand side, the solutions we have for viruses and vaccines. As said, in the therapeutic protein space, predominantly hormones, as well as, in this case, factor 10 or albumin for albumin fusions. For antibody types, for virtually antibody format, right, whether it's an FC, through our FCXP, Kappa XP, Lambda XP, CH1XL, but as well for the more exotic type of antibody formats like IgE derived or IgM or IgA derived. On the right-hand side, our solutions for viruses and vaccines, as said, predominantly now launched as GMP, the AAV product lines, but we are working on adenovirus, as well as lenti, as well as exosomes in that space. So really a snapshot of what we have in our pipeline. Again, we do have a custom service, but if you are working on any of these targets, then feel free as well to reach out, because we always like to collaborate with our customers to move these products through our pipeline. So that was, in summary, our platform which we've developed, right, from work package 1 up to work package 5, to end up with, let's say, a GMP suitable product. And I would now like to turn to a case study, and this was a co-development with Genexine, a Korean company, to make a capture select product which would need to be specific for TSH, right? So that's thyroid stimulating hormone. And the request here was that it shouldn't be cross-binding to any other hormone of that gonadotropin family, and should improve the process efficiency and cost, of course. And what we actually did, we worked together with Genexine and went through our work package 1, through our work package 5. In this case, I'm going to only focus on, let's say, the latter stages of the program in course of time. But this is actually where we picked it up in work package 3, right? So as you can remember, in work package 3, we have about 6 candidates. In this case, showing the data of 3 of them, which were TSA ligand 3, 6, and 7. And this was actually tested by ourselves and by Genexine. And we demonstrated in this case with pure material that although all 3 ligands actually performed, we could clearly see that TSH3 had some issues with elution, which you can see on the left-hand side by the strip fraction of TSH3. And TSH6 and 7 actually, showed good capacity and efficient solution. What we then did was actually test with feedstock. So we tested the same ligands, but then with feedstock, and demonstrated that TSH6 and 7, although both worked, still TSH7 had some material in the strip, and TSH6 actually gave full elution. So finally, moving into work package 4, we selected TSH6 as the final candidate to move into let's say final manufacturing. And these are some of the data of that specific product, showing the dynamic binding capacity testing of candidate 6 on the left-hand side, showing a pretty high capacity for this hormone, so almost reaching 15 grams per liter at a 5% breakthrough, which is pretty good for a molecule of this size and class. And on the right-hand side, the purity analysis, showing that on sec HPLC, the purity is let's say higher than 99%, with a recovery of about 63%. So finally, giving the conclusions of the performance, comparing it to the old process. So the old process, of course, was a multi-step process, having poor binding capacity and strength, poor elution, moderate purity, and a low recovery. And by using candidate 6 in the middle lane here in this table, we were really able to improve the purity, efficient elution, the binding capacity, and as well the final recovery. Improving, let's say, the cost of goods by and reducing that from let's say 46% to around 71% in terms of cost saving. So again, successful collaboration with Genexin, and thanks to them for them as well in helping us, let's say, develop this specific product for TSH. So that was a case study on the Capture Select affinity technology development. Besides actually starting from scratch and defining and developing an affinity resin, making as well the affinity ligand, we as well offer custom resin development when actually the customer has their own, let's say, affinity ligand or affinity product. What we then use is our, let's say, our core porous backbone. And for those of you who are not familiar with the porous backbone, it's actually based on polystyrene divinyl benzane as a backbone. And the uniqueness here is that you've got a really nice linear pressure flow curve. Because it's a rigid bead, you have linear and scalable performance as well as easy handling. It is highly robust and chemically stable. Through their large through pores, you have reduced mass transfer resistance. So your capacity and your resolution will be very well maintained over a wide range of linear velocities, which really truly makes porous unique in its attributes as composed to, for example, an agarose-based backbone, which in the end can result in more efficient purification. On the right-hand side, you can see an electron microscopy picture of the 50 micron bead, which provides, because of its size, superior resolution, as well as improved capacity through some novel surface chemistries which have been developed for, let's say, ion exchange and HIC, finally resulting into excellent pressure flow properties, while as well, of course, being let's say, fully scalable in the Bedford facility. So our custom development program for porous follows basically, let's say, the same process as our Capture Select process, but of course with less steps, right? So because again, in this case, we assume that the customer will have their own affinity ligand. And what we start off here is we start off again with the work package 1, which is called prototype resin development. Initiated, of course, by technical discussion with the customer, finally ending up in an experiment joint design, a proposal, and the final result of this work package 1 is prototype resins, which can have a fast turnaround of around 6 to 8 weeks. We would then move into work package 2, which is final process definition, where we'll do process optimization, a resin formulation, the quality assays, and the scale-up testing. And as a final step, we will move into work package 3, which is the commercialization, which is again is making these products, let's say, suitable for GMP processes. Again, taking a similar approach as with our Capture Select resin development by minimally using 3 by 5 liter batches. Of course, it can be higher. As well, at that point, initiating stability studies. And finally, similar to what we do for the Capture Select custom design program, is developing regulatory support packages and COVs. So a very, let's say, bespoke and comprehensive custom resin development program. And in this, let's say, prototype resin development, we can tune specific parameters. So, of course, we'll be using the porous bead, but we can select different pore modes based upon the size of the molecule target. We can differentiate in the coating chemistry, which we have within our toolbox in porous. Play around with the linker length, the coupling chemistry, the ligand chemistry, as well as the ligand density. So these are all aspects which can be tuned in this porous development program. So to make it a bit more clear, I want to present here a small case study on a resin we've made, which was a custom porous carbohydrate resin. The application in this case was a depletion, so basically a polish of a product-related impurity for a clinical manufacturing process, which needed to go into basically manufacturing. And in this specific case, there was a product-related impurity, and the request of the customer, because the current commercial resins basically lacked throughput and base stability, to come up with a product which could be used basically to move into clinical and final manufacturing. So we worked in this case together with the customers to do the prototyping, the scale up, and the manufacturing of the validation batches of this specific custom carbohydrate resin. So if you take a look to the left-hand picture here, the first thing we started off with was, of course, affinity ligand screening. Although the customer had an affinity ligand, we as well checked 2 other vendors for that specific affinity ligand. And 3 of them were actually screened. And ligand 1 was selected based on performance, but well as on availability and cost. As you can see, ligand 2 actually performed less in terms of, in this case, contaminant log reduction, but ligand 1 as well was, from an availability and cost-wise, the most attractive one. Second, on the right, we did ligand density and efficiency testing. Again, on the left-hand side of this right graph axis, you can see the contaminant log reduction. And on the right-hand side, you can see your coupling efficiency. Of course, coupling efficiency, very important for your cost of goods of the affinity resin, because that determines how much ligand you actually would need as a function of the ligand density. This graph clearly shows that at, let's say, 3 mg per ml, we had, let's say, the highest coupling efficiency, equal contaminant log removal as the 5 grams per liter ligand density, and outperforming the 1 gram per liter ligand density. So the 3 mg per liter, the 3 grams per liter ligand density was selected based on maximum clearance and highest coupling efficiency, giving as well, let's say, the best contaminant log reduction of around, let's say, 3.5 to 4 logs. What we further did, as said, as well with poros, we can tune the pore mode, and we can check coupling conditions. So what we did here from the left-hand side is we checked coupling conditions to actually see, okay, what is, let's say, the best coupling condition, varying ligand concentration, buffer conditions, reactant time and temperature. And coupling condition 1 was actually selected for scale-up, which is the one on the left-hand side. In the middle, you can see the 2 pore modes we selected for this specific product. So 450 angstrom and 2000 angstrom, and checked a contaminant log reduction. What you can clearly see here is that 2000 angstrom had a lower ligand density even, but showed a higher and significantly higher contaminant log reduction. So bumping up the contaminant log reduction from about 4 to 8 logs. So the 2000 angstrom resin was actually selected. And finally, of course, we checked, let's say, cleaning and stability in 0.5 molar sodium hydroxide. Again, demonstrating that the ligand and the resin itself is stable for 150 hours with a contact time of 0.5 molar caustic. And the log reduction was maintained over this 0.5 molar of caustic. So finally, within this program, we started off with, together with the customer, selecting the right ligand, selecting the right process to make the affinity resin, selecting the right pore mode, as well demonstrating the log removal. First, the identified resin candidates was produced at 500 ml scale to confirm scalability, and all process parameters were set. And as a final result, we made the 3 by 5 liter scale validation batches together with the regulatory support documentation to be able to use it in a clinical manufacturing process. So just a short summary on the different services we offer for custom resin design. So as I've explained, in our toolbox, we have got our Capture Select Affinity Ligand and Resin Development Program. In this case, we'll start from scratch by identifying the best suitable affinity ligand, which will be tailored to your specific product. We can immobilize those affinity ligands on 2 different backbones, which are in our standard platform, which is either agarose-based or porous-based. So we have got those 2 options, which are again built-in choices within our custom development program, within our capture select technology, or we can use our porous chemistry and products and use a ligand which has been supplied by the customer. Finally, in, let's say, both cases, ending up with a fully validated resin, which will contain a regulatory support package in the case of the capture select technology leakage ELISA, which can as well be and which can as well actually have as a benefit that you have additional analytical tools. So with that, I hope that the presentation has demonstrated that whatever challenge you have in purification, within our toolbox, we've got the necessary tools and upscalable tools to help you out in a milestone-based collaborative development process, really working hand in hand with you to ensure that you get the right product. That we are an integrated supplier, which can work with you from R&D prototypes to resins, which can be incorporated into GMP, finally at larger scale. And that we have a track record, right? So we're not doing this and ended up with a nice, let's say, prototype. Now we've got a track record of really developing these products from scratch and ending up in suitable, let's say, GMP processes. And with that, I'd like to thank you for your attention, and I'd like to open it for any questions you might have.
Okay, and as a reminder, you can go ahead and type your questions in at the bottom, and we will ask those now. So the first question is, can you support us until the lead screening before prototype resin testing?
Sorry, then I need to, can you support us until the lead screening before prototype testing? Yes, absolutely, and that is as well something we have done with customers. So we, of course, always like as well our customers to test the product themselves, right? So usually we would deliver prototypes to the customer, but we as well have worked with customers who were at very early stages where we even have made, let's say, reference material, right, in the case of some of our products. So it's case by case dependent. Of course, you know, we'd rather have it that the customer as well, let's say, confirms and tests. But absolutely, we've done that before.
And what kind of regulatory support is provided with a custom resin?
Yeah, so that's as well a great question. So we really make no distinction between a custom and a, let's say, general use product. So if we make a product, then yeah, basically we do all of the identical steps for a custom resin. You, as part of the custom development program, will finally, in the end, will get a regulatory support file, which is identical to what we have available for all of our other products. So we absolutely make no distinction. We as well will develop the leakage ELISA as part of our work package 5 to ensure that that's being developed. So we really make no distinction.
And what can you say about leachable ligand issues with agarose-based custom platform resin, cleaning, sanitization strategy, and resin cycles?
Yeah. So yeah, there as well. If you look to ligand leaching of any of these products, they are similar to what basically is out there for protein A. You still are immobilizing, let's say, a ligand onto a backbone. So in any event, you will always have some ligand leakage, and that is similar to what is observed out there in the industry for protein A type of products. For any of the products, of course, we as well, will work together with the customer to find suitable cleaning, let's say, strategies and regimes for these type of products to ensure that, of course, you can get the maximum cycles out of this product as an affinity resin. That will, of course, finally depend as well upon the feedstock which is being used, which can have, let's say, an impact on cycling. And of course as well on the final ligand attributes, right? On what strategy needs to be used. But again, as said, this technology is already being used at commercial scale, right? So again, 300-liter columns are being run with this technology for, let's say, cycles which are, feasible at that scale.
And is it possible to combine multiple targets into one project?
Yes, so as well, something we developed a couple of years ago was to take, for example, a customer pipeline and build a library of that. So we can include up to 10 targets in one library construction to ensure that, at the point that you need an affinity ligand, you can remove about 10 weeks in a screening program, because that would basically be the waiting time to get the right immunization and to be able to build the library. So yes, we can definitely do that, and we can do that up to around 10 targets.
And is there a way to use identified ligands or custom resin for analytical purposes?
The identified ligands, and that is as well something which we offer in our Capture Select, product approach, is that once we get into work package 3, so basically after work package 2, we have a subset of ligands, which are basically the 15 kilodalton single domain antibodies, and they can absolutely be used in analytical settings, right? So you can take that ligand and then basically after biotinylation, use it on any streptavidin platform, for example. You can use it in octet systems, gyro systems, biocore systems, or everything actually which has a streptavidin, linker to it. And that is as well a pretty big advantage, right? Because from that point on, you basically already have an analytical tool available as well, which we can then guide your further process development as well. Yep.
Okay, I think we have time for just one more question. If we didn't get to your question, we'll be passing those on to Dr. Siekstra, and he can follow up with you directly. But if you still have any questions, please don't hesitate to enter them now, and we will pass those along. But the last question for the webcast is, what is your experience with custom resin manufacturing in the vaccine space?
Yeah, so in the vaccine space, again, I mean, what we've done there actually is we've developed a really, nice technology which is called CTAG. And that is a 4 amino acid C-terminal sequence, which you can actually use and which you can leave on in clinical and final manufacturing. So this is really truly a platform for vaccines, and specifically for recombinant vaccines, because that actually means that for these pretty, sometimes highly complicated products, you can leave the tag on. And there's actually some very nice papers out there by the Jenner Institute in Oxford, who have used this for the malaria vaccine, and actually demonstrated that they could nicely and seamlessly move into clinical manufacturing by leaving the tag on. So although, of course, it is possible to specifically develop something for vaccines, a first question we would always pose if we get customers for a custom program is basically say, do you really need it, or are you actually better off perhaps by incorporating the C-tag as a C-terminal tag in your product, because that might already actually do the trick and give you a platform for let's say your recombinant vaccines.
Okay, well, thank you, Dr. Siekstra.
Thank you very much.
Thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.